Steve has been for a long time a pioneer and major contributor to single molecule biophysics. And I was thrilled that Steve was willing to give this talk, because I've heard him talk many times over the years, all of which have been memorable in some way. I think this will probably add to that. And then I realized it's kind of caused a bit of a problem, because what was I really going to say about Steve to introduce it? So I could either go on for way too long telling stories and telling everything, all of his accomplishments, and then by the time he actually talked, there would be an angry crowd. So I decided that was probably not the best idea. But I thought in looking through Steve's CV, I would highlight some of his contributions and his honors, and then he can speak for himself about his science, and I'm really looking forward to his talk. Steve was a graduate student with Howard Berg at the University of Colorado. He was a postdoc with Jim Spudich at Stanford. That's where I first met him. He was just down the hall from me, and he would always bring me a cup of espresso when he wanted to ask a complicated question. So there's some sort of calculation in there right from the beginning. After leaving this postdoc at Stanford, he worked at the Roland Institute and at Harvard, and then was at Princeton from 1994 to 1999, at which time he moved back to Stanford, where he's been ever since, as a professor in both the departments of biological sciences and of applied physics. Steve's had many, many honors, and I only want to highlight a few of these. He is a fellow of the American Physical Society. He won the Max Dill Brook Prize in Biological Physics from the American Physical Society. Selected to the National Academy of Sciences in 2007. He was a member of the Aspen Center for Physics. Steve has established and continues to organize the biannual Aspen Conference in Single Molecule Biophysics. He also has had another role as an advisor to the government on biological weapon threats, and has worked for many governmental committees in that area. Steve has given somewhere around 60 keynote and plenary talks in the last 20 years, and I'm real pleased that he's willing to give another one here tonight. As a member of the Biophysical Society, Steve won the Young Investigator Award in 1994. He was elected a Fellow of the Biophysical Society in 2009. He's been on the Public Affairs Committee. He served as president. He has been a chair of a subgroup. He was the program chair for the annual meeting in 1999. He's been on the nomination committee. He served on the board and on council, and has had just a large impact and has devoted a lot of time and effort into the quality of the society and its annual meeting. Now, Steve has another great talent, is that he's a musician. He's a award-winning bluegrass banjo player who told me that he decided that banjos were too big and he switched to mandolin more recently. My main memory of this is I remember back at Stanford, where every day around noon he used to take his banjo out and practice right under the window of the administrative assistant of the Department of Neuroscience, who was not pleased at all. Not that that had any effect on Steve. And that I believe he's not going to demonstrate any of his musical talents tonight, but I'm really looking forward to, and I hope you'll really enjoy his scientific talk. Steve?
Everybody hear me? All right, I'm live. Well, it's wonderful. It comes with another plaque. I've got a whole collection, as you heard from now by the Biophysical, and an award check. We actually spent this last night going out to a nice restaurant, so I thank the Society for that. I have returned to the scene of the crime. The very, very first Biophysical Society meeting I ever went to in my life, it turns out, was in 1983, and it was here in San Diego. And that is, in fact, the last time the Society's had its meeting in San Diego. And it was a life-changing event for me, and so it's a special thrill to come back to San Diego after all these years. It's a sobering realization that was 29 years ago, and there are graduate students here who weren't even born when I went to my first Biophysical Society meeting in San Diego. That meeting was also punctuated with a side trip to Tijuana, accompanying a colleague who was on a quest to buy a conga drum. Now, I can assure you that most people go to Tijuana are not after buying that. So that was an interesting adventure, and it reminds me that biophysics is filled with unusual characters, and I see myself surrounded by them here at the meeting. So today's talk is shedding light on single molecules. I will shed a very little light on the subject because it has become a vast area, and I won't be able to do proper justice to it, but I hope to give you at least a personal perspective on things, and we'll see how this develops. But before I start, I should point out that like many of you, I've suffered rejected papers. And we've all gone through this experience. And having gone through recently the experience of 4 rejected papers without benefit of review, I thought I'd share with you by way of commiseration what is arguably the worst favorable review of all time. I sent this out to Ed Egelman, in fact, because he's now got this behind him as retiring from Biophysical Journal. But before I share this with you, I have to teach you a little vocabulary for those of you who don't know it. The word lucubration. A lucubration is basically a midnight raving. It's from the Latin lucubrationem, or nocturnal study, or lucubrare, meaning to work from artificial light. And the review goes as follows. I have searched the feeble lucubrations of this author without success for some trace of ingenuity, acuteness, or learning that might compensate for his obvious deficiency in the powers of solid thinking or of calm and careful investigation. This manuscript uncovers no new truth, reconciles no contradictions, arranges no anomalous facts, suggests no new experiments, and leads to no new inquiries. As this paper contains nothing which deserves the name of either experiment or discovery, and as it is in fact destitute of any species of merit, it should certainly be admitted to your proceedings to join the company of that multitude of paltry and unsubstantial papers which are being published by your journal every month. The good news is he says publish, okay? You gotta go for the bottom line here. Now, this review was written in 1803 by Henry Brougham. His name was pronounced Brougham at the time. He was very famous. He was a baron. He actually founded University College London and eventually became Lord Chancellor of England. He was a publisher, and it was in fact Edinburgh Review where he was anonymously publishing these reviews, even though he owned the journal. But he was a scientist. What's perhaps more striking, as many of you may not realize, the paper that he's criticizing Thomas Young's wave theory of light. Now, for those of you who don't know, Thomas Young is one of my heroes. He's arguably the original biophysicist. Proto-biophysicist Thomas Young died in 1829. He was a true polymath. Thomas Young, as many of you know, contributed, of course, to the wave theory of light. There's Young's double-slit experiment showing interference. He's also the Young of Young's modulus. For those of you who, for example, calibrate your suction pipettes in neuroscience, you use the Young-Laplace equation for surface tension to find out the pressure. He's responsible for the Young-Dupré equation for surface wetting. But that's just on the physics and engineering side. He was also an MD, a doctor. He saw patients. He's responsible for the modern three-color theory of human vision. He actually was the first to describe the fact that the lens was responsible for accommodation, and he's the first person to talk about astigmatism. Doctors know him for inventing Young's rule for child doses, which is how much drug to give a child if you knew the adult dosage, and is responsible for studies of hemodynamics. In music, he invented a new kind of temperament, different from equal temperament, called Young temperament. And He was also a linguist. He's fluent in over 15 languages. He wrote an Encyclopedia Britannica article in which he described over 400 different languages he happened to be personally familiar with. The word Indo-European language is a term introduced by Thomas Young into the lexicon, and he proposed the first international phonetic alphabet. As if that weren't enough, he was the guy who helped decipher the Rosetta Stone. So when you look around at your fellow biophysicists here, and you think of these as accomplished people, remember Henry Brougham and remember Thomas Young. Oh, he was involved in other things. You know, back at the time, of course, the French and the British really got into it about issues, including the Rosetta Stone and who was the first to decipher it. There was also this discussion of how long the meter should be, and as many of you know, it's part of it's 1/10,000th of the meridian, or the half meridian from the pole to the equator. But at the time, they were trying to figure out what a good way to measure this would. Thomas Young suggested something very interesting based on what Christopher Wren had first talked about, which was to make it the length of a pendulum whose period would be 2 seconds. So the clock would, every time it went tick, tick, it would tick at 1 second intervals. And in fact, that's very close to an exact meter. It's within less than 5% of actually less than 0.5% of the actual length of a meter. But of course, it varies a bit with longitude, with latitude rather, which is a bit of an issue. He also was one of the first people to get involved in life insurance, which is not such a bad thing these days. All right, but on to the topic at hand. I'm here tonight to tell you about single molecule biophysics, a subject that's near and dear to me. And single molecule biophysics is obviously a fascinating area, and I might have been born to do this because, as many of you know, my full name is Stephen Michael Block, so my initials are SMB. So I'm here to tell you about SMB. It's a wonderful field, and it's really been a revelation because many, many years ago, I would come to a Biophysical Society meeting and I'd perhaps see 1 or 2 or 3 posters in this area, and now today the mean free path between posters that do single molecules is measured in distances of meters. So it's obviously an up-and-coming field. It's been very wonderful, very powerful. And the idea, of course, is to look at things at the individual level and not have to worry about the details of ensembles and averages. And there are many different analogies one can express for why this is so. I'll give you a couple of my favorite. For example, one is, imagine you're sailing a ship from New York to San Diego. If the ship is sufficiently small, you'll pass through the Panama Canal and come up the other side. If you're not a Panamax-labeled vessel, then you have to go all the way around Cape Horn, maybe skirting the Malvinas or the Falklands, depending on your predilections. But if you consider the average path taken by a ship between these 2 points, it passes through Peru, Carlos. It actually passes also through the Atacama Desert, the driest desert on the planet. And yet this is supposed to be a waterbound path. No ship, in fact, takes this route. And so if you were stuck with ensemble averages, you might be a little bit misled. My favorite analogy, though, was put forward by Sandy Simon, who pointed out that when you consider humans on the average, we have 1 brain, 1 heart, 2 lungs, 2 kidneys, 1 ovary, and 1 testicle. So consider here a gel, as it says here, the workhorse of biochemistry. This is actually a runoff gel where we're watching the length of RNA as it's being transcribed by RNA polymerase. It gets longer and longer, so the bands go from heavy from short at the bottom of the gel all the way up to heavy and long at the top of the gel. And the lanes are taken at different times. And you notice that the gel has different bands at different places. That's because it's going along base pairs at a time. But also some bands are dark and some bands are light. The problem is that the darkness of a band here is related to the probability of stopping at that point times the lifetime of that point. So you don't know whether a lot of the molecules stop for a short period of time or a small number of molecules stop there for a long period of time, because all you can learn about is the product of those. And this is akin, if you will, by another analogy, to looking at, say, a marathon race from a high altitude, and you might be able to figure out where the water collection stands are, and you might be able to get some sense of where it's uphill and downhill. You can't learn as much as if you could, say, study someone like this. Here's Robert Choyot, the 4-time winner of the Boston Marathon. This is the sort of thing that we really like to look at. And of course, today, as many of you know, it's possible, for example, to look at a single enzyme, in this case RNA polymerase, transcribing DNA. It moves along it, and you see a rich record filled with more or less constant velocity, but punctuated by a whole bunch of stops. So it moves and fits and starts. The flat parts are the stops, and they have various different durations. And those pauses are there for a reason. They're there to help to regulate genes and do interesting things. So our lives are enriched by being able to look at things at this level. My life has been enriched by being exposed in my academic career to what I call some singular role models. These are the people who helped make me the scientist that I am today. We all have our favorite heroes and mentors, and these are some of mine. I want to acknowledge in public my debt of gratitude to all these people. It was Max Dill Brook who found me when I was a young undergraduate at Oxford and brought me first to Cold Spring Harbor Labs and then to Caltech and turned me into a biophysicist. Max actually gave the national lecture in 1970, the year after he won his Nobel Prize. He then literally sent me to work for Howard Berg. I was instructed to go work for Howard. And then ironically, when Howard left the University of Colorado, he came back to Caltech, where I finished my PhD in more or less the position that Max had occupied before he retired. And then years later, optical traps were invented by Arthur Ashkin at Bell Labs, and I came to know him, and he gave me some apparatus and some tips, and it's been a real pleasure, and I'd like to acknowledge each of these special individuals. And in terms of singles, they have single things to say. Max Dill Brook, as many of you know from the famous Dill Brook Luria fluctuation test, taught us that single spontaneous mutations are the things responsible for evolution. Howard Berg worked on single bacteria and showed that they can make decisions and that the individual flagella rotate. And Art Ashkin was responsible for showing that single particles and even single atoms can be trapped and studied using light. And single atom traps have since been used to achieve things like Bose-Einstein condensation, for which Phillips and Steve Chu, amongst others, won the Nobel Prize. So there's a lot of great science that has come out of all these individuals. And I'm going to tell you a little bit about that, folded into this rather personal story of mine. This is going to be a personal perspective on my path through single molecule biophysics. So let me apologize in advance if I leave out a lot of people who are in fact here tonight, whose contributions to the field are equally strong or stronger, and have wonderful things to teach us about biology and how molecules work. But I'm going to look at the history here through the soda straw of my own existence. And I'm going to take you back, in fact, to the point where I think, in my perspective, single molecule biophysics really began. And that began back in the 1970s. And curiously enough, not only did single molecule biophysics get its start in the 1970s, around that same time I was getting my start, thanks to Max Dill Brook, who brought me out to Caltech. Shown on the left is a picture about 30 miles from here, actually, in the Anza Borrego Desert. Max liked to take his people on desert camping trips. He was legendary for that. In the background there is Toby Dill Brook, who's today a professor of neuroscience at ETH in Zurich. And there's my famous camper, Ludwig van. And my wife and I had adventures that we can tell you about over a drink on that. And shown on the right is the Long Beach Bluegrass Festival in 1975. So I was just getting my start in science and having a blast. I was at Caltech that year. Around that same time, in the early 1970s, Art Ashkin was thinking about how lasers affect matter. Now, the laser had only been invented in about 1960 or 60 to 64. It was really undergoing development then. So the laser wasn't even quite 10 years old. But Art Ashkin was interested in the fact that you could get very intense beams, and he was interested in what radiation pressure would do to things. And he shined laser light into cuvettes, for example, into water where there are spheres. And this is an actual hand drawing that Art made at the time. And what he noticed is that the beads would get sort of pulled into where the center of the beam is and then shot along the beam and pushed all the way to the end. This was not an optical trap, but it was a demonstration of the fact that light could induce radiation pressure, and those forces were quite prodigious. They could affect micron-scale objects. He wrote down a little diagram, which was later to become famous, pointing out that the light on the center of the beam got deflected by the sphere because it acted like a little lens, and that deflection produced more reaction momentum because it had greater photon flux than the light that was at the periphery of the beam, which is so the one in the center beam is labeled A, the one at the side is labeled B. So A has more photons than B, so A produces force A, Fa, which is longer than force Fb, which is shorter. The net result is the thing gets pulled into the center of the beam. And this propensity to do this turned out eventually to be the principle by which optical traps would be built, but they wouldn't be built for another 15 years. He did, at that time, do the cover of Scientific American, though, And shown here is a sphere which he was able to levitate in air by arranging the laser. He eventually built a feedback circuit that would stabilize it so he could keep a sphere sort of floating in the air using this centration on the beam effect. Also, the early 70s was a time when Howard Berg started studying the biophysics of bacteria. He got into this partly because through the work of Julius Adler at Wisconsin, and Berg, as many of you know, built one of the first tracking microscopes. He followed bacteria as they moved up chemical gradients and discovered that, lo and behold, they could make decisions, they could increase their run lengths when life was getting better, they could run, they could tumble. And he even proposed, based on some cross-linking experiments, that the flagella that drive bacteria literally rotate like the prop on a submarine. That turned out to be right. And then he spent some time collaborating with Ed Purcell, and he wrote some very famous papers. I don't think you can even call yourself a biophysicist unless you've read a copy of The Physics of Chemoreception, arguably one of the most important papers ever published in the Biophysical Journal. Some of the ideas from that paper made its way into Berg's little book, Random Walks in Biology, and I know many of you have this on your shelf. It's a wonderful book. He eventually wrote a book on bacterial motion. And this is the guy who was sort of got me really started along my path at graduate school. So I got to thinking about that. And he got me excited by one particular experiment. This experiment was done in 1974. It wound up in 3 back-to-back-to-back papers in Nature, cited here on the right. And this was the tethered cell assay. It turns out you could stick down a bacterium by its flagellum, and the tail would wag the dog. And so when the motor tried to turn, it would turn to spin the body of the cell around. So you didn't have to use indirect cross-linking experiments. You could visualize it directly, and you could get records of when it ran one way and ran the other, and it turns out clockwise rotation turned out to be tumbling, and counterclockwise rotation turned out to be running. And I'm going to show you a video here, which I recorded way back when the Earth's crust was still cooling. And this is the experiment that really got me excited about biophysics, and I've been doing things ever since, charged up by this particular experiment. These are tethered cells. If you look at the one in the yellow circle, you'll see that it turns in both directions. It reverses at random. These are Poisson statistics. And you can measure the time constants for either of these. And for my thesis work, in fact, I wafted chemicals over these individual motors and watched how they responded as a function of time. And so you could use this to tease apart the adaptation mechanism in the chemotaxis apparatus and a bunch of stuff like this. But this, as it says, was the mother of all single motility assays. Here was, for the first time, was the output of a single molecule or molecular complex resolved in real time in a way that was palpable and that could get you excited and that you can measure, quantitate, and model. Now, today we know a lot about the bacterial motor. Here's one of these beautiful EMs that David DeRosier is responsible for, and some pictures from Kenichi Namba's lab that show what we know about the motor today. It's, of course, run by the proton motive force, a current of protons. It's about 45 nanometers in diameter. It's big as motors go. It's the biggest known motor at 11 megadaltons. It's run on by about 0.2 of a volt that the metabolizing bacterium charges up its membrane with. It goes pretty darn fast, about 300 hertz, and in some forms that run off sodium, it'll even go up as fast as 1200 hertz. That turns out to be not that much. It's only about 60 femtoamps of current that are driving this. Prodigious torque in the range of 3,000 piconewton nanometers, and H-force generators. And more recently, using some optical trapping methods, Richard Berry and his colleagues have actually finally been able to resolve the individual steps that it takes as it rolls around. There are approximately 26 of these, although that's still a little bit controversial. But this is an example of the kind of biophysical information you can get if you can study single things. And when I was working with Howard Berg, I did an experiment that turned out to be my first step experiment, and I've been looking at steps ever since. What we did is take the mode B protein, which is responsible for torque generation. It's one of 2 proteins that do that in this motor. We put it under lac control, and we added IPTG to a tethered cell, and for the first 20 minutes or so, nothing happens, and then suddenly the cell starts spinning and gets faster and faster and faster. And if you blow up what happens just as it starts, you discover that it starts not smoothly, but abruptly, and goes up to a certain rotation rate. And you sit there watching for about 30 seconds, and then its speed suddenly precisely doubles. And the reason there are little layer lines there is we were collecting the data on video, and so they're separated by 30ths of a second because of the video records. And here's another record. Sometimes it would go a little slower, and then back up again, and back up again. It would get faster and faster and faster, but it would get faster at equal increments of angular velocity. And if you plotted the rotation rate against second level, you've got these beautiful straight lines. And of course, the long cells, which had more viscous drag, would turn slower. They give lower slopes. And the shorter cells would go faster. If you normalize them all by the drag, all the lines would now suddenly fold onto the same value. So what does this tell you? This tells you that these force generator units get incorporated into the motor one at a time, and each time they do so, it leads to a uniform step in torque. And eventually David Blair was to do these experiments in a more controlled way and show that there are about 8 torque generators all in this motor. So these were my first steps. That got me hooked, and I've been going ever since. Right around that same time that the tethered cell in 1974 was developed, and by 1976, Erwin Neher and Bert Sackmann were getting started on some work that, as many of you know, got them the Nobel Prize in 1991. Here they're shown today, rather bald. Back in 1976, they looked sort of still less hirsute. Actually, this was taken somewhat later. This is about 1990. Back in 76, Erwin Neher had more hair. But they published a landmark paper on the observation of single-channel currents. The first single-molecule observations of any single proteins were arguably single-channel records done in electrophysiology. And I'm putting this up from their paper just to point out that where they acknowledge the work of Chuck Stevens, who played a really important role in the early development of this and spawned many great scientists from his lab who went on to look at this. Of course, as you all know, they discovered that if you fire polished an electrode and you pushed it up against a cell membrane, you could, under some conditions, achieve a really tight seal, the gigaseal, a seal whose resistance was on the order of a gigaohms or so. And back in those days, with a mere 500 megaohm feedback resistor, they could get records of the tiny amounts of current that would flow when channels open and close. And here, in the technology of the day, they literally took a photo of the oscilloscope, and this is what you see. There's a scale bar for, you see several picoamperes of current and several levels that the channels are opening, and these are, of course, the acetylcholine channels that are found in muscle. Here are some records from that first paper with 3 different agonists. This is what really spawned the field. It's been going ever since. These were absolutely fantastic. And if tethered cells didn't get you excited, this ought to. Eventually, today, of course, now we have structure here from Nigel Unwin from a review, is the acetylcholine receptor. And the records got better and better. By 1983, they were starting to look this good. And these were collected in the computer of the day. It's hard for me to even give some of the young people perspective on this. They're collected on a computer called the 1134, and I see many of you nodding your heads, and all of you who are doing that have gray hair. The PDP 1134 was before the microprocessor came along. It cost between $12,000 and $25,000 in 1976 dollars. I don't know what that would be in current dollars. Contrasted, if you will, with the laptop I'm using for this presentation, which is a tenth the price. The CPU was really 2 giant boards and used this famous thing called the LSI-11 chipset, which is a computer implemented in 16 bits. 16 bits, by the way, can only address 64K of memory. Whereas the 64-bit CPU in this thing is 1 chip. The address area, as I said, was 264K against 256 terabytes. Clock speed, you could, depending on the price you're willing to pay, you could get as fast as 15 megahertz. But you could only get 256K of memory. My laptop has 6 gigabytes. My little USB drive has 16. And the storage. Oh, the storage was on 8-inch floppies, something called the RX-01 floppy disk. There's a picture one. But don't worry, if you got those filled up, you could get an RQ-05 hard disk that was about this big around, held a big 2.5 megabytes. So we've come a long way. This being the Cold War, it was cloned by the Soviets, and they had something that would run the same instruction set called the SM or CM1420, and there's a picture of that. All right, so then came this landmark book, this very famous book, Single Channel Recording. That's actually my copy there on the left. It's now in its 2nd edition. Hey, Rick, I think you've got a chapter in there, don't you? Yeah, several of the Biophysical Society people have this, and this was a Bible for me back in the early days. It's a book that still has amazing currency. But I was, looking through Nature the other day, and there's the cover of Nature just this month. Channels are still very important; they're still with us. Optogenetics is being featured at symposia here, and the structure here of a channel rhodopsin was recently solved and found its way to the cover of Nature. So channels and the ability to see these and the ability to record from them directly have really galvanized single molecule biology. Now, fast forward a little bit to the time of the early 1980s. As Rick said in the introduction, I did my postdoc with Jim Spudich, who also was a national lecturer here back in 1995. And Jim convinced me to come to his lab because of the experiment that you see here. In fact, he sent me a reprint, and this is the reprint where he signed his name to it. And I made a deal with him. You've heard from the introduction that I brought coffee around. I said I would come to postdoc with him only on condition that he would provide space in his lab for my cappuccino machine, which he did. Mike Sheets took some time off and went out to work with Jim Spudich for a year, and they came up with an assay. It was the mother of linear motor assays. It turns out there's an alga. This is Nitella axillaris. That's what Jim looked like back then. And this alga does cytoplasmic streaming. And if you look at it carefully, you can see vesicles being moved. This is in real time. The fastest myosin in the world is not in a hummingbird. It's not in you or me. It's in a plant. This plant myosin will move at about 100 microns a second. Compare that with 6 microns a second for my muscles, and you realize how impressive this is. But it turns out if you have very, very tiny scissors and you're willing to cut your way up the cylinder of the plant, you can flush away the cytoplasm, flush away all these vesicles, and reveal the actin cables, which are serving as highways for this transport that run along the tops of all these chloroplasts. And so you see there a picture from their paper with the chloroplast rows, and now what you could do is take small spheres which you've coated with myosin, say from a rabbit, and put it down on the actin cables, and if there's ATP in the buffer, away they'll go. And you see in the picture on the right, the little fluorescent spheres in yellow, and you have a time-lapse picture here, so you see at various different times the progress of a yellow sphere against a row of red chloroplasts. They're red because chloroplasts are naturally fluorescent anyway, so you put it in the fluorescence microscope, and this is what you see. So this was a door-opening experiment because now you could look at myosin motility completely in vitro. And so when I was a postdoc, I did an experiment which turned out to be, in some ways, to presage a lot of other experiments in which we later learned how to hook motors stereo-specifically to beads. We didn't use beads at the time. We actually used bacteria. We used Staph aureus because, as many of you know, Staph aureus has protein A on its surface that binds an antibody. Great. You can use it for an anti-myosin antibody. Great. So you could use that to hook up a myosin motor. So we hooked up a myosin motor, and that worked. But then we got fancy, and we hooked up a fragment of the myosin motor, in this case something called short, heavy marromyosin. This is a fragment of the myosin motor which is actually missing something called the S2 region. And that moved too. And why was that interesting? Well, it had been proposed that a helix coil transition in the S2 region of myosin might be responsible for its motion. Well, if it still moves and you've chopped that part out, that part's probably not responsible for the motion. So this experiment told us that the model that had been proposed by William Harrington at Johns Hopkins was probably not the right explanation for myosin. And of course, today we know a lot more about how this molecule really works. Now, while this was going on, but not quite in time for me to finish my postdoc, Art Ashkin was getting busy, and he was finally developing an optical trap worthy of the name. And in a very important paper published in 1986, which Steve Chu was a co-author, he observed finally a single beam gradient force optical trap, and this is the optical tweezers that we all know have come to love today. They were actually looking at how it worked from the side. They weren't actually using the microscope lens they used to make the tweezers to view the thing, which in retrospect makes me surprised. I don't know why they did that, but this is the way they hooked it up. This is the apparatus diagram from their paper, so you can tell from that it's really hard to reproduce this experiment unless you know what you're doing, because the illustration is not much of a guide. But they talked about something called negative light pressure due to the gradient force. And here's the actual picture from the paper. Once again, it's very hard to sort of see from this what's going on. But if you're looking at something trapped from the side, trapped in that focus is a sphere, and what you notice about it, if you look really carefully, is that the angle of light that emerges from the sphere going down is actually narrower than the cone of light that comes in from the top. And so I've shaded in some sectors to try to give you a sense of how that angle changes. And that's responsible for this negative pressure, which in turn is responsible for an optical trap. So optical trapping works sort of like this, at least in the ray optic regime. Everything I'm going to say to you I'm going to take back in a minute, but this is a good way to think about it. You can think of the bead as a spherical lens. So when you send light through on a microscope objective and it goes through the bead, if the bead is moved off axis, it'll tend to bend the rays. And as a result of bending the rays, you're sending the momentum off in a different direction. So by Newton's third law, there has to be an equal and opposite force, which acts through the center of the bead. In this case, if the bead is displaced to your left, it'll feel a reaction force to the right, restoring to the center. By symmetry, on the other side, the reaction force will also bring it to the center. If if the sphere goes beyond the focal point, it'll tend to focus the rays forward, and they'll carry now more forward momentum than they did coming in. As a result, there is negative pressure, or negative radiation pressure, tending to push back into the focus. This is what they were referring to in the abstract. Likewise, if you were to push the sphere a little closer, it would now spread the rays out, and it would feel force going forward. The net result of all of this is there's a restoring force tending to pull the bead into the center. So an optical trap is nothing more than a three-dimensional spring made out of light. It's a spring whose spring constant can be calibrated with exquisite detail, and it offers a lot of opportunities for experiments. Ashkin was no slouch, and he immediately got started trying to look at whether he could trap things. And he was using an argon laser at the time. And he went down the hall at Bell Labs and got some bacteria from one of his friends and some tobacco mosaic virus and other things. Actually, he didn't get bacteria. He got tobacco mosaic virus to start off with. And then he wrote a paper. And once again, in the paper, he showed the apparatus. It's the same drawing, which also makes it very hard to figure out what's going on. But he wrote about it in the paper in language that you just don't find written anymore, and I wanted to read this to you today. He said, in most of our experiments with silica colloids or tobacco mosaic virus in water, we noticed the appearance of some strange new particles in diluted samples that had been kept around for several days at room temperature on the bench. They were quite large compared to Rayleigh particles on the basis of their scattering of the light and were apparently self-propelled. They were clearly observed moving through the distribution of smaller, slowly diffusing Rayleigh colloidal particles at speeds as high as hundreds of microns a second. They could start up again, and frequently reverse their direction of motion at the boundaries of the trapping beam when they encountered a dark region, indicating some sort of attraction to the light. Their numbers increased rapidly as time went by. This is a physicist discovering life. So he had neglected his sterile technique, and his tobacco mosaic virus specimen was getting more and more contaminated as time went on. And so sure enough, he found himself trapping bacteria when what he set up to do was to trap viruses. He was looking at the 90-degree scattered light signal. Every once in a while, the needle would just go off scale. But the problem is then the needle would come crashing down again because the bacterium would vent its contents into the medium. And Art Ashkin coined the word, which we use to this day, opticution. So the problem was opticution, and how do you get around that? And Art was no slouch. He realized that, well, I've got to change the wavelength, and he went to a near-infrared beam at 1,064 nanometers. There's almost a kind of window of transparency for biological objects. They tend to absorb water when you get out to too long a wavelength, and of course there are colors and chromophores and pigments that absorb it in the visible, so the near-infrared's a good place to be. And he built something like this. And I asked him once, why did he use a negative lens there? And he says, oh, that was just for the drawing. I actually had rigged up a different way. I had it going in through the eyepiece of the microscope. Oh, you did? Anyway, it was on the basis of this drawing that I went out and developed my own optical trap shortly thereafter. And this appeared in 1987. Optical trap, as many of you know, is the closest thing to a tractor beam that man has produced. It is, in fact, nothing more than a diffraction-limited focus. And here is what happened, or probably was presumably something that's similar to what Art Ashkin saw when he first trapped a bacterium. Here are E. coli swimming around. I knew how to grow these. And now we're moving the beam of light around, and we control the horizontal, and we control the vertical. This is all in real time. The scale is, you see the 5-micron scale bar, so the screen is about 20 microns edge to edge. And we can exert complete three-dimensional control over the life of this poor bacterium. It's a rod-shaped object, so it aligns with the beam because the optical trap is weaker in the vertical direction than it is in the transverse direction. In a moment, we went down to the surface. Here we see it at the surface again, where some cells are stuck. Some of those are even tethered and turning around. We can move it about. And then, of course, if we shutter the beam, the cell swims off, untrammeled by this experience, and we don't have opticution to worry about. There we go. So that was an optical trap. And I was at the Roland Institute at the time. I built the trap within about 6 or 7 days and was often doing experiments very quickly. What I wanted to do was take these tethered cells and spin them around. Experiments which I'm pleased to say are still going on today. I even heard a talk at the meeting about a very similar kind of work that's continuing to this day. You could spin it around if you're willing to spin an external lens, and it turns out what you're twisting is not so much the flagellar filament, which is stiff, but a little connection which connects the flagellar filament to the shaft of the rotary motor. That thing's called the hook. It's 55 nanometers long, consists of about 120 subunits. So this wasn't quite a single molecule experiment. This was a 120 protein experiment. But you could twist up that hook and you could measure its torsional stiffness, and you could show it had all the properties that you wanted for a universal joint so that you could couple power off axis. It had been proposed to work in this role, and here was finally quantitative evidence that it was working that way. And if you look carefully in the diagram, you can see going into the microscope this long arm, and that was because the arm was perched on a lens and moved by magnets. We wanted to steer the beam around. So, shown in panel A, yeah, you can move lenses, but if you're smarter, you build a confocal microscope. Many of you know those are built with galvanometer mirrors. That's another way to move beams around. You can move the source with fiber optic cables, or if you really want to get fancy, you use acousto-optic modulators, which let you move light with microsecond rise times. And Kuhn Fischer, who was a postdoc in my lab, was one of the first to pioneer the use of acoustic optic modulators in moving optical traps. And shown here is a little demonstration where you could move the trap to different positions, and by time sharing, you could produce up to 10 traps at once. Not to be outdone, a former postdoc of my lab created eventually the world's smallest game of Tetris. The world's smallest game of Tetris is contained within a square about 50 microns wide. It uses multiple traps to trap a number of beads, and here it goes. It can be played in real time. This is falling down. This is another shape that formed. You can rotate it around. You can dock it. This is obviously an advanced player here. He moves it over, leaving good space in anticipation of the square which is to come, thus clearing out 2 rows and allowing the game to continue. All right, I won't bore you with this, but thank you. Thank Joost van Memmeren, Theo Pialaga, Brougham van den Broek. Okay. We wanted to study single motors, and one of the things I got involved with was Kinesin, which, as it says here, is nature's tiniest mechanoenzyme. Of course, neurons in particular have a big problem with transport because they can grow up to meters long, like the drawing shows you, and diffusion won't do the job, so you need something to transport it. The cells, of course, have evolved this network of axoplasmic transport. There's retrograde and anterograde transport. Kinesin has turned out to be the motor discovered in 1984 by Ron Vail, Mike Sheets, Bruce Schnapp, Tom Rees at Woods Hole. And that motor moves at microns a second, and it can transport things along arrays of microtubules. Yeah, if you're unlucky enough to get an orthopox virus like smallpox, it'll actually hijack your kinesins and use them for other purposes. If you have bad kinesins, then you can get some nasty diseases. And of course, kinesin is also a target for anti-cancer drugs. Shown here is an electron micrograph of some cargo and a putative kinesin motor on a microtubule. And here's a little film, not from me, but from Howard Hughes, who has lots of money, and from the inner life of the cell, showing a lot of the things that we've actually learned in the years over kinesin. It's a motor that is small, it moves hand over hand, it moves vesicles. Notice that the paths taken by each of the 2 heads are actually different through space. There's kind of a left head path and a right head path. They're not the same, but the step sizes are the same. And here it's moving along inside the cell, which is remarkably free of things. And here it is pulling Jabba the vesicle behind it. So real kinesin doesn't work like that. It works with Brownian motion, but this shows you many of the characteristics that we were able to learn by single molecule experiments. Here's dimeric kinesin. It has 2 heads, a stalk, a tail. The structure of the heads is known thanks to the work of Eckhard Mandelkal and colleagues, and there have been several structures since. And John Kuhl was kind enough to give me this overlay, which shows that the structure of kinesin can be wholly contained within myosin. If you will, it's a kind of a subset of the myosin molecule. They're superposed here on the ATP, or ADP, which they both bind. Although they seem to work by slightly different principles. In particular, both heads of kinesin can bind ATP, but unlike myosin, the ADP state is the weakly bound state in the case of kinesin. We know from single molecule work how processive it is. We know the dimers take steps, how big the steps are, how often they occur. We know from single molecule experiments about the fuel economy, which is on everyone's lips these days as prices of gas go up. This takes a single ATP per 8 nanometer step traveled. And we also know from single molecule work that kinesin takes what's called a so-called asymmetric hand-over-hand walk, and the 2 heads are able to coordinate somehow. So back in 1990, I got involved with optical traps and kinesin, and we built an assay which sort of recapitulates, as it says here, what nature herself does. Instead of hooking an organelle up to the molecule, we took one of these beads that we've been talking about, hooked it up to a kinesin molecule. And with an optical trap, you could work at such low densities of kinesin that you could take a bead which only had 1 kinesin molecule on it, and normally it wouldn't interact with microtubules in a month of Sundays at random. You could move it to the microtubule, force it to interact, release it, and watch it move. That's what you see in the in the little photographs in the bottom right. And this was the first optical trapping bead assay. Shortly before this, Joe Howard, in a seminal experiment conducted with Ron Vail, showed very beautifully that single Kinesin motors can move in gliding assays microtubules. So here was a motor in which one was all it took. So this was a single molecule biophysicist's dream, because Kinesin is a motor that works in singles and not in multiples. So we wanted to learn more about it, and we built what we called the optical trapping interferometer. This was a device which was actually inspired by Winfried Denk. It's another example of the cross-fertilization between the field of electrophysiology and the field of single molecule biophysics and, on the motor side. And he had built a sensor for looking at tiny displacements in hair cells in the ear, and it was based on the way a Nomarksky microscope generates contrast. It was a very clever device, and we realized that his device would not only work to detect displacement, but we could also put an optical trap through it. In fact, if we were really clever, we could use the same light that was doing the optical trapping to do the distance displacement, and there in one device we would have something that not only trapped, but gave us a readout of displacement at very high resolution. So the optical trapping interferometer was born from the phoenix of Winfried Denk's detector. So these days, of course, we don't even bother with that anymore. We have very fancy detectors that use what are called duolateral position-sensitive detectors, or PSDs. Other people use quadrant photodiodes. And we learned to do a fancy kind of what's called back focal plane detection, which is immune from certain types of noise and drift that the earlier apparatus was not, was subject to. Here's a demo film. It was put together, I think, by Josh Shavitz in the lab, showing a Kinesin bead being put on a microtubule, and then you see these little runs. And so this is Kinesin-powered motion, powered by a single motor in the presence of ATP. And this is the sort of thing you can now study. So I did. And so this is the second step paper in my life. We discovered that Kinesin moved in discrete 8-nanometer steps. And these correspond to the spacings of the dimers on the microtubule. And it was published back in 1993. And this is the modern version of that experiment, a little demo video put together by Ewan Andreasen. And what you can see here is that the motor is starting to move. You can see its small movements based relative to that spatial bar, which is not moving. And every time it moves, you record in real time the steps. As it gets farther out of the trap, the resistance gets tighter, and you can start to see the steps a little better. You even see some back steps. You can see stalls, and you can see starts. You get a lot of data in very short time this way. It turns out that high stiffness means lower noise, and that's really good for people in my field who work in this area. By the equipartition theorem, there's a half kT of energy in every degree of freedom. So if this thing's on a spring, you could solve in one line for the mean square displacement. It's just kT divided by the stiffness. And now the stiffness can be complicated because it's stiffness due to the motor itself and also to the trap. But if you make the trap really stiff, that can drive down the mean square displacement. And if the motor stiffness is nonlinear and you can drive into a regime where that gets stiff too, you have a double win, and now you can get down into the regime where you can watch displacements that are really quite small, smaller than you might have guessed. So how do you get the force up there so that you can stretch this spring? And the next thing to do was to develop the optical force clamp. So this was developed by Kuhn Fischer and Mark Schnitzer in the lab, and this is basically a trap follower. The bead would move, as it shows here, and then you'd follow behind it a certain distance with a trap in a feedback loop, moving it forward every time the bead moved forward. And the trap would, in this case, would be 175 nanometers behind. And as you can see here, you could keep the force constant to better than 5%. So this allowed you to study Kinesin as a function of a control parameter, in this case force, and move it up and down, learn lots of stuff. Here are some Kinesin steps under a force clamp regime, and now you can begin to see the steps really quite well, each of them beautifully resolved. You can look at power spectral densities of these. You can get another decimal place. 8.2 nanometers is the average step. And as I said before, that corresponds precisely to the spacing of the tubulin dimers in the lattice of a single microtubule, which are spaced actually about 8.15 nanometers apart. I'm going to fast forward to today, since we only have a certain amount of time tonight, and say that as a result of a great number of single molecule experiments over decades of work, we're finally, I think, very, very close to writing down the complete two-headed pathway for Kinesin, understanding how one head affects its contralateral head, what states ADP and ATP binding it goes through, who moves when, with what rate, where the load-dependent steps are, where the ATP-dependent steps are. There's a pathway for forward stepping. There are two pathways for backward stepping. There's a pathway for just throwing away the energy and doing ATP feudal hydrolysis. And all of those are shown in a diagram like that. There was actually a poster up on this today. If you missed it, you really missed it. We have achieved this level of understanding, in fact, thanks to a great number of experiments, all brought to us courtesy of single-molecule biophysics. So I tend to take at this point a motor-centric view of biology. Not only can you look at enzymes like traditional enzymes that are mechanoenzymes like kinesin and dynein and myosin, and this meeting's been full of reports with exciting developments on all those three things. But there's also, of course, the processive nucleic acid enzymes, the polymerases, the ribosomes, the helicases, and they're all grist for this mill. Shown on the right are some experiments that various people have done, lambda exonuclease, RNA polymerase. The ribosome, what I'm fond of calling the Mount Everest of motor proteins, has been studied by Carlos Bustamante and his colleagues with some very elegant experiments. ATP synthase, the rotary motor that's responsible for ATP production. And the list goes on and on. I'm particularly fond of the little icon on the lower right of the flagellar motor. I got that from a creation scientist website. If you ever want to get some really good pictures of flagella, you could go to Google Scholar and look up David DeRosier, but you might as well just look up creation scientists. They've got it all out there. It's good, and high production values, great graphics. Do it. All right. I want to tell you a little bit about our optical trapping studies of RNA polymerase. That was the next thing we got into. We needed apparatus that was even more sensitive to study this, and so we got to do this. We built an apparatus when I moved to Stanford. It was built into a clean room. It was noise isolated. It was temperature controlled, vibration stabilized. All this because, as I'm fond of saying, when you build something like this without meaning to, you've built a seismometer, you've built a thermometer, and you've built a microphone. And to show what a good microphone it is, Josh Shavitz, who was a student in the lab at the time, took a CD player in the room and played some music, and then was recording the position of one of these beads stuck to a glass surface. The bead is executing nanometer scale motions, and a Brownian motion, basically. But it's picking up a tiny microphonic from the noise that's in the air. And if you now play back our position signal as a function of time as a wave file through the computer, you can hear the music. So I present to you the world's most expensive low-fidelity recording. Some of you might even recognize the tune, if you're old enough. Now the hiss in the background is Brownian motion. Where's Jose? Yeah. Brazilian. All right, enough of that. Stop. Okay, very good. So we studied RNA polymerase. We studied the bacterial version in collaboration with Robert Landick. As many of you know, this is the enzyme that reads the genetic code. It opens up a transcription bubble by prizing apart the two DNA strands. By protein standards, it's big. It's not as big as the flagellar motor. It's not as big as the ribosome, but it is nonetheless thousands of amino acids. But it's only 10 nanometers across, so this is true nanotechnology. This, of course, reads a DNA template, synthesizes an RNA, and it's really the nexus of gene control, as it says here in bacteria. There are over 100 different factors that interact with this one enzyme to modulate transcription, which is quite impressive when you consider the size of the molecule and the availability of binding space. And it's been studied for many years. A lot is known about it. As biologists had referred to three distinct phases: initiation, elongation, and termination, which from a physicist's perspective are nothing more than start, go, and stop. And the rest is just vocabulary. But we've been studying mostly the go phase, but recently we've been starting to look at the stop phase and even the start phase. It's a complicated enzyme with a complicated life cycle, and I'm not going to take you through all of this, but not only can it add bases to the RNA, but it can also slide backwards along the DNA, extruding the RNA from one of the entrance pore where the bases come in. And this is a sort of enzymatically dead state because it normally wants to add onto the 3' end of the RNA, but it can't. It can pause in response to certain signals that form in the RNA, for example, hairpins. It can do what's called ubiquitous pausing. About every 100 bases or so, it'll take it into its own to pause anyway. It can, in the presence of high pyrophosphate, actually chew up and knock out a base that had recently condensed called pyrophosphorylasis. It's a smart enzyme which can recruit factors when it detects errors in front of it and get them fixed. And of course, it has to know how to terminate, and one form of termination also involves the formation of a hairpin. And so our lab has been studying a lot of this. I'm not going to bore you with this, but there are lots of papers here of using single molecule techniques to approach each and one of these different phases. We use an assay which we've called the dumbbell assay. It involves 2 optical traps and molecule DNA stretched between the two beads, held in the traps with a molecular RNA polymerase. So here it is, optical trapping for dumbbells. This will give you a sense of how the experiment actually works. You're going to see 2 things, an optically trapped bead at the top, and then one of these dumbbells which is stuck on the surface in the bottom. Now we're in real time, and we're using those acousto-optic deflectors now to move this bead around. We're going to use that to calibrate the system. And while that's being calibrated, you can see the larger of the 2 beads in the dumbbell is stuck on the surface, and the smaller one is dancing around it because it's hooked up by a DNA tether, and it's doing Brownian motion, and it's like a mad dog on a leash, and it can't get anywhere. Okay, so now we're calibrating the trap. Now we're going to zoom in on that, and you can see how this is actually done. As we raster scan through the photodetector area, we accumulate an array of offset signals. This is used to fit to a 2D polynomial, which is then used to linearize the whole thing so we can measure position to within a nanometer or even a fraction of a nanometer over a detector area of about 100 and 150 nanometers on a side. So now we're going to grab one of those dumbbells off the surface, and that's done by suspending it in the 2 optical traps. The other stuff's out of focus, it's down below. And now we're going to take one of those beads and stretch the DNA molecule, which of course you can't see. Here we are stretching it again. We can use that stretching to tell us whether we have a single tether, a single DNA molecule, or more DNA molecules stuck between them. We do this by measuring the persistence length, and this tells us whether we in fact have single molecules. The one thing you should always ask a single molecule biophysicist at the end of his seminar is, how do you know you have one molecule? Some of them are not prepared to answer that question. All right, but we're ready to run the experiment here. And here, this molecule has RNA polymerase. This bead has RNA polymerase on it, and we're looping the film. It's sped up 30-fold so you can see it, but it's clearly moving along the DNA and making the tether shorter. We can measure that position record as a function of time, and you can see, again, these pauses and other things. It's very, very rich in data. One of the things we wanted to do, which after we'd seen steps in Kinesin, after all, was see if we could see steps in this molecule. It had long been known that RNA was synthesized 1 base pair at a time by this enzyme. What had not been known was whether it moved along the DNA 1 base pair at a time, or as suggested by some individuals, Mike Chamberlain, for example, at Berkeley, whether it pulled in a bit of DNA a bit at a time like a jib sheet on a sailboat and moved in a series of discontinuous elongations. So Will Greenleaf and Elio Bonazzieri and Josh Chavis in the lab got involved in this experiment and of trying to hunt for individual steps. The instrument had to get really good for this. One of the things we discovered is you send the laser beam just through the lab. If you weren't careful about it, the beam would fluctuate. They twinkle for the same reason the stars twinkle in the sky. It's that you get density fluctuations in the medium, and those density fluctuations produce gradients of refractive index, and that acts like a grin lens and bends the beam. So shown here in red is what happens in air. That's the power spectrum of our instrument. When we go to helium, particularly the low frequency end of the spectrum gets a lot better. And of course, the integral under that is giving you the integrated noise power. So in the lower graph, there's the 1 angstrom line, and you can see that this apparatus will achieve a sensitivity of better than 1 angstrom from really quite low frequency out to about 50 hertz. So here was something that could measure the position of an enzyme to within better than the diameter of a hydrogen atom and make that measurement 50 times a second. So this is just what we needed for these experiments. We also needed another trick, and this was invented also by Elion Will, and Michael Woodside had a hand in this. This is a passive force clamp. We wanted to be able to clamp the force. You already know about how the active one works with feedback, but feedback's too slow. You've got to measure position, you've got to take an average, you have to feed back to your computer. It turns out if you pull the bead out into the nonlinear region of stiffness in the trap, where momentarily the force extension curve goes flat, the stiffness there is 0. And the position of the bead is related to the position of the RNAP through the equation you see there. And if that stiffness term goes to 0, hopefully you can all solve the equation in your head. Did I do that wrong? This should be over the other one. How about that? My mistake. Anyway, you can't solve that equation to get this, but it turns out the x bead is delta x RNAP, and this allows you then to measure the displacement with very high accuracy and high bandwidth. And so you get gorgeous records that look like this. Shown in pink is the noise. Shown in black is the signal. You can't even see them make them out. So you blow it up by a factor of 10, and now you can see the noise envelope and the signal averaged over some time base. But you're still not seeing steps. Now we're going to blow out by another order of magnitude, and finally now each of these grid lines is drawn 3.5 or 3.4 angstroms from the next, and you're starting to see individual steps. We see more records like this. The rungs of the DNA ladder are separated by intervals of 3.4 angstroms, and so here was finally an apparatus that could resolve individual molecular steps. We were very excited about this. And it wasn't just a tour de force of measurement, it also, by measuring the force-velocity relationships and other things, it could tell us a lot about how RNA polymerase worked and where in the cycle motion was, and it told us that this enzyme probably worked as a kind of rectified Brownian ratchet. We learned a lot from these experiments. And so going to higher sensitivity is always a good thing if you're a physicist. The ultra stability imparted by this passive force clamp also allowed another set of experiments, which are pioneered in the lab by Mike Woodside. But the original experiments of these types were done by Jan Lippard when he was working with Nacho Tinoco and Carlos Bustamante. They did some beautiful work on looking at the whole folding and unfolding of RNA hairpins. We started to look at DNA hairpins using this dumbbell trap, a simple variation of what you already saw. And as you can tell from the cartoon, if you're pulling at the bottom of a hairpin and unfolding it, the extension of that provides a sort of natural reaction coordinate, which allows you to follow the progress of the reaction. And shown here is as titrated as a function of force in the force clamp. Mostly at high force, the hairpin is open. Mostly at low force, the hairpin is closed. In between, it does something else. And all the electrophysiologists in the room are saying, gee, that looks familiar. I've seen that. Change the holding potential, and this is what the channels do. Well, this is the mechanical analog of a lot of stuff that has already been worked out by electrophysiologists who came before. And that made life very pleasant for us. Here's the unfolded probability. Surprise, surprise. It follows a Boltzmann relationship. And you can measure very accurately from things like this the average opening distance of the hairpin. You can measure the so-called F1/2 or unfolding force value. That's a distance, that's a force. The product of the force and times the distance is an energy. And in fact, you can get the free energy of folding from this and compare that, for example, with results you might get from calorimetry. And this is really a very accurate way of returning these kinds of energies. But you don't have to stop there. Michael Woodside proceeded to do an entire zoo of hairpins, looking at what happens as you change the length, the loop size, you change the barriers, the heights of the barriers, the GC content, put in mismatches, you name it. Thanks to the high resolution of this, we were eventually able, in fact, to reconstruct an entire diagram which showed by iteration the potential energy diagram for folding and unfolding of the entire hairpin. The one that's shown there actually has an intermediate state because there's a mismatch, so it'll fray up to the mismatch. That's a metastable state, and then it'll open up the rest of the way. So this allows us to study nucleic acids in a new way, and we moved up in complexity then to the next thing. We wanted to look at folding actually as it occurs off of RNA polymerase. So the holy grail here is to actually understand co-transcriptional folding and how that actually works. And so we then started working on riboswitches, and Kirsten Frieda and Will Greenleaf did some nice work. This is the PBU-E riboswitch. Many of you know about riboswitches already. Ron Breaker's at Yale done a lot of really great work on this. They're non-coding RNAs that selectively bind small ligand molecules and then control gene expression. And they can control gene expression through transcription. They can control it through translation. There are even riboswitches that control, in eukaryotes, alternate gene splicing. So they control things at various different levels. The one we're going to talk about here controls transcription. And this is an adenine riboswitch. It binds adenine, forms a little aptamer that binds that. That aptamer uses up some RNA. If the adenine comes off, some of that same RNA can go into forming a terminator hairpin, which is actually more stable than the aptamer itself. If the terminator hairpin forms, then RNA polymerase falls off because that's the termination signal, and so the gene will be off. So the gene will be on or off depending on whether adenine folds or not, and it actually works as a kind of kinetic race because all of this is happening co-transcriptionally. The structure of this has been solved, shown on the right as a structure, and shown as a sort of secondary layout, color-coded on the left. And the assay we used to do this was one similar to what you saw before with the dumbbell, but now we're going to attach a handle which is going to hybridize to the end of the RNA. And so we catch it as it emerges from RNAP. We're going to let RNAP transcribe a free-floating template until it runs into a roadblock and stops, at which point it's made something. And this is a very general assay. You could use it to make a ribozyme, a tRNA, a riboswitch, a hairpin, anything you like. You can code for it, and now you've got it hooked on both ends. You've got it hooked on one end by the polymerase and the other end by a handle. You can pull on it, And here are some poles. This is these days being called force spectroscopy. It's got a great name. And you can fit these to worm-like chain states. It has a folded and unfolded and clearly an intermediate state. And you can tilt the energy landscape by putting in various different forces and see how this behaves. And in particular, there's a remarkable theorem many of you know about due to Chris Jarzynski. It's arguably the most important result in non-equilibrium thermodynamics of the 20th century, which shows that you can actually use diagrams like this, where you measure the work, which is the area under the curve. You measure the work out of equilibrium while you're pulling this, and believe it or not, from that non-equilibrium measurement, you can actually find out what the equilibrium free energy is. You have to do a funny kind of average where you exponentiate things, and that requires your data to be very good and very stable, and that's a whole talk on itself. But if your data are good, you can actually use it to get that number. And you can also use these data to do something else. You can ask at what force do they start to unfold, and what is the force unfolding distribution like? And due to some other remarkable work that Gerhard Hummer and Attila Szabo and Olga Duka are responsible for, you can model this as a non-equilibrium motion over a barrier and get the barrier height. So not only can you get the equilibrium free energy, you can get the barrier height, you can get the barrier position, you can learn about the energy landscape involved in the unfolding of this aptamer. And from this, we were able to derive that the energy barrier has a certain height. More interestingly, the offset to that barrier was about, as this shows here, about 2 nanometers. And that pointed us to a particular base that was in the bottom of the bottom loop, the P1 loop. And it turns out a lot of riboswitches actually have a GC base right at that position, sort of like a snap, to snap the whole thing together when the thing folds. So this was a piece of sequence that was brought to our attention simply by doing a single molecule biophysics experiment. But you don't have to stay out of equilibrium. You can also look at equilibrium, set your force clef to some value, watch it flicker in and out of position. Here it's open, and it flickers momentarily to the P2 folded state. That's great. Now we can reduce the force. Now it flickers to the P2 folded state and a third state. Let's reduce the force again, and now we see other states begin to appear, and again, until we're all done. And now when you worry about the different offsets, because they're all at different forces, you can discover this is a 5-state system. It goes from fully unfolded to P2 folded to 2 P2 and P3, and then it's competent to bind adenine but hasn't bounded yet. Then it binds adenine. And from all the relative populations of these, and from the force dependencies of the transitions, and all these data, you can actually reconstruct an energy landscape for this entire system without adenine or with adenine. Here's without adenine. We see what folds first and in what order. And these are the kinds of diagrams you see in PChem textbooks. But in those PChem textbooks, they're all notional diagrams. They say reaction coordinate, and they say relative energy, and then they have some graph that somebody drew off the top of their head. Here we're finally seeing a real free energy diagram with error bars and real scale bars, and extension becomes our reaction coordinate. And of course, if you add adenine to that, it stabilizes the adenine bound state, and that's what drives the aptamer switching in this riboswitch. So in unpublished work, and I'm taking this opportunity to go out on a limb here, I'm very excited to report that we've also been able to watch co-transcriptional folding. So if you're making this thing and it gets longer and longer, say you're making a hairpin, as you go through that hairpin, nothing folds until you start coming down the backside of the hairpin, and then it starts folding more and more. And if the forces are right, that thing will not get longer, it'll actually get shorter. So here you see an actual data from the transcription of a hairpin. It gets longer and longer until it makes the loop, that's in blue. And then once the loop is made, it starts to try to transcribe the backside of the hairpin, and now it starts coming together and looping up, and it gets shorter and shorter until it finally folds. But now transcription continues, so it gets longer, and then it finally reaches a roadblock. So since you can do that, here are some actual data from a real experiment that Kirsten Frieda performed in the lab. And what you're seeing now is a trace of a full riboswitch. Now it's got the aptamer, and it's got the expression platform, which in this case is the terminating hairpin. And P2 folds, and then the terminator folds, and then the whole thing gets released. But if there's high adenine present and you have just the right amount of force, you can also see traces that look like this. In this case, P2 folds, but then the rest of the aptamer, P3 and all of the rest of it, come together. That prevents the terminator from folding, and so then it goes all the way through the terminator position, hits the roadblock, and then it's too late, but now some other structure, a little bit of structure forms, and it gets a little bit shorter. And so you can actually predict, based on traces like this, whether the trace is going to run through or terminate simply by looking in the middle here find out what's folding and what's not. So this is an example of folding telling us the fate of the molecule, and it's an example of being able to look at co-transcriptional folding in real time. This is very exciting for us. You can build up an entire sequence of what happens. RNA polymerase moves until it gets down to the first point where P2 can fold. Then, if the aptamer can fold around these positions, it will. And then, in some cases, the terminator hairpin will begin to folding, and then it will release. But in other cases where the aptamer is stable, you don't get termination, you get run through, and it runs through to this position. And so you can build up a picture like this of the whole fate of it simply from single molecule experiments. So we're very excited about what we've learned about this. Along the way, we've also done another riboswitch. This is the eukaryotic one from thiamine pyrophosphate. I won't tell you about it tonight, but this was just recently published in PNAS. So we have now another landscape to compare, and this is from a riboswitch that occurs in eukaryotes, in this case in plants. So where is single molecule stuff going? Well, lots of different places. And if you've been around the meeting this week, you've heard fabulous talks using fluorescent techniques, which I didn't even touch on tonight. But I can tell you that the optical trapping people have been, are quite knowledgeable about these areas, and vice versa. And the fluorescence people are building their optical traps, and the optical trapping people are building their fluorescence instruments. And the instrument of the future will combine all of these things. Shown on the left is some work from our lab on the next generation instrument, which will do fluorescence from a single fluorophore and trapping. Shown on the right is something from the work of Jan Chemla, working with TJ Ha, which uses time-sharing traps and also does fluorescence and single molecule trapping. To put it in perspective, there's 15 orders of magnitude more light in an optical trap than the tiny bit of fluorescence you get from a single fluorophore coming back and giving you fluorescence. So this is the signal you're chasing down, but it's possible to do this. And so I think this is the future of the field. We'll have molecular probes within the molecule that allow us to learn about what's moving. And we'll have forces at external control parameter to be able to move it and test it and do all the other things that we can do with optical traps. And so I think the future is very rich and very exciting. So I want to thank you for your attention. It's been a long time. But before going, I want to thank the people that made this all possible. With the wonders of PowerPoint, I bring to you every postdoc and grad student who's been through my lab, and 3 of my most important collaborators. A lot of these people are here tonight, in fact, about half of them. It's a terrific group of people. They've built the instruments, they've done the experiments, they've analyzed the data. I've had a long collaboration with Jeff Gellis at Brandeis, who was at grad school with me and got me into single molecule things. With Bob Landick at University of Wisconsin, who's responsible for a lot of our RNA polymerase work. Steve Rosenfeld, who's here, and we've done some nice work on Kinesin together. And all these fabulous people, and these are the biophysicists who make it all possible. So I thank you for your attention, and I thank them.